Bio 100.
Okay, welcome back. As you guys saw in the first video, funding for science is actually really critical and can have ramifications way past individual research projects. And while the example previously was about differences in wanting to look for cheaper ways to do diabetes treatment versus kind of basic microbial research, the era of kind of having these just kind of general questions and trying to get them funded is kind of coming to a close. As noted at the end of the video, funding for science is actually really, really competitive these days. And as a result, researchers now kind of have to act as salespeople, finding ways to sell the different funding agencies the merits of their research. So something, if you stay in the sciences, that you will hear time and time and time again is, what's the question? Meaning, what are you as the researcher proposing to study? Not what do you want to study, but specifically what are you going to study? There are big differences between kind of broad, nebulous research questions and a clear question with a well-rationalized reason behind asking and wanting to answer that question. And so we're going to get into that a little bit right now. So if you're going to be selling your research or proposing your research, something that really matters is who you're trying to sell it to. And so if you're in the US, some of the major research funders are mostly government entities. They are, by and large, taxpayer-funded entities. So not only does your scientific question need to be interesting, it should be something that not only is interesting scientifically, but is something that eventually might give something back to those taxpayers or society as a whole. That doesn't mean your questions need to be directly applicable to society. Indeed, the example video we started with was looking at basic research on microbial defenses, kind of paved the way for advancing our knowledge, and in a way kind of moved us forward and opened more doors that eventually did kind of pay back those taxpayers in all sorts of different ways. And so to get right into it, how do you think about finding a good question for funding agencies is really oftentimes the same process as finding a good question just as a researcher, finding a good research-related question. And so this process is not going to be much different from the scientific process or scientific method that you learned about in some of your earlier biology, where you're really going to be focusing on observation. Using that observation to come up with a question in biological in nature, and maybe taking that observation in question and generating a hypothesis. Eventually, you're going to predict or think about what types of data would allow you to address that hypothesis. So, what data might you need? And then, if you had the data, how would you analyze it? And then say you got the ideal data and performed the analysis and got the output you expected. What would those conclusions, what kind of conclusions would you be able to draw related to your original question? And so this is kind of the general process. Make an observation, find a way to make it a testable hypothesis, and then think about in kind of the ideal world, what kind of data and how would you analyze it in order to address that hypothesis? And if everything worked out just right, what would you actually get out of it? All right, so that's kind of the general workflow. And so let's be a little more specific, because specificity is really important in science. So let's say we have a question. Oftentimes it's going to be comparing something to another thing. That's kind of how we often do science. And so really you're going to want to define your kind of comparative groups or populations. This could be ecological in nature. Maybe you're thinking about comparing populations of individuals of one species in a given area. So maybe individuals in the forest, maybe deer or something, versus individuals that live in the desert. And so, this could be population A and this could be population B. You might think about populations as being different strains of some bacteria or influenza isolated from patients in New York versus California. It could be anything. It doesn't have to be a population in the sense of how you learned about it in intro-bio, interbreeding individuals. It just basically needs to be a well-defined subset of a larger thing. Very rarely in science are we sampling and comparing the whole of everything. If you're looking at the impact of the forest versus the desert on deer herbivory, for example, you're not ever going to measure all the deer in all the forests and all the deer in all the deserts. You're going to be looking at maybe deer in the Klamath forest, something like that, versus the Mojave Desert. And I don't know how to spell Mojave. Maybe that is it. I'm sure you guys will let me know. Anyways, so you're going to be subsetting the whole into a digestible piece, and we call those kind of our groups or populations that we'll actually be doing the science with. So once you've identified what your population or groups you're going to be comparing, and you might have a sense, like we talked about herbivory between forest and desert deer. Now we want to really define our variables. What exactly are we going to be measuring? In other words, what kind of data are we going to be collecting to answer our question or comparison? We've talked a lot about variables so far, whether they're categorical or continuous or independent or dependent. And so all those things are important to note. A variable can be any factor or trait or condition. It can exist in different amounts or types. It could be something like length or quantity or temperature or speed or mass or distance or depth. It just needs to be something that varies, and you're interested in whether it varies between your different groups or populations in different ways. The word vary is actually in the word variable. Variables vary, or at least you hope they do. Ultimately, what you want to do is use different combinations of these two components, your composition of your groups or populations, along with the variables, and use these two things to design your experiment. How many populations are you going to be comparing? How many variables are you going to be varying across those populations or measuring? And so the interaction of these two different things, groups and variables, allow you to kind of address three broad categories of kind of research questions: descriptive, comparative, and correlative. And we'll go into these a little more in depth in the next couple slides. So the first one I want to talk about is what many people refer to as kind of a descriptive investigation. This is often involves describing or quantifying part of a natural system. Often you're only going to have one population or group. So we've only got one population or group. And we're only going to be measuring one thing within that group, one variable. And so this is typically not a statistical comparison. This is something that we're trying to describe something we observe in nature. It could be something like, how many pine trees are there in the Mammoth Hot Springs area? What is the wolf pack distribution range? Or how many different species of wasps occur near the Riverfront Cafe? The next kind of broad type of question or experiment is what we would call correlative research. Again, we're going to stick with just one population. But now, rather than looking at a single variable, we're going to be comparing three different variables, or two different variables, or five, but more than one. And these could be things where you might be generating a linear regression because you might be interested in the relationship between two variables within this population. Something like, what's the relationship between the length of the tail and age in a population of humpback whales? Or how does a spider's reproductive rate change with a change in season? Or how many grams of salami in your lunch as you're sitting at the Riverfront Cafe impacts the duration of wasp visitation? So you could look at the correlation of any type of variable 1 versus variable 2. We looked at a population of cars, for example, several weeks ago, and looked at the correlation between two different variables, weight and miles per gallon. And so that's a correlative type of research question, and you can do that with just about anything. Okay, and so the third type is more of what we would call a comparative research question. And this is kind of the opposite of correlative. Here we're looking at a single variable maybe something of importance like body length or the diversity of fungi in a given area. But now we're going to measure that variable that was originally part of a descriptive study, and we're going to look at it across multiple populations. And those, again, could be populations in an ecological sense. You could look at the amount of grass eaten or herbivory of deer would be your variable. So grass eaten. And whether that varies across the desert, the forest, or, I don't know, the ocean. And so here you're taking a descriptive study and comparing it across 3 different things or 4 different things. They do not have to be these ecological situations, though. It could be across different treatment groups. A treatment group could be a type of population, different strains of bacteria, or bacteria grown on different media. Those are all different populations in a sense. But where you're measuring the same variable across multiple groups or populations, we would call a comparative research question. Examples of this could also be, is there a difference in body length between male and female tortoises? You could think of tortoise sex as being a group or population. Is there a difference in the diversity of fungi that live in a forest compared to non-forest areas? Oftentimes, in really well set up studies, one of these populations, particularly with related to where you're looking at treatments rather than ecological populations, one is going to be a control, where maybe you had no salt in your media and you were comparing the differences in concentration of salt on bacterial growth, something like that. Or someone sitting at a lunch table with no grams of salami would be your control if you're looking at grams of salami influencing wasp visitation or something. All right, so hopefully that gives you kind of a sense of the general things that you should look for in a question and identifying what you're interested in looking at, and then identifying what your groups or populations and your variables are, and using what those are to decide whether it's going to be a descriptive study, a correlative study, or a comparative study. Or maybe going into it knowing, hey, I need to do a comparative study. So what kinds of variables and what kind of groups can I split out in order to build a testable question that will be comparative or will be correlative or will be descriptive? What you're ultimately going to want to do is observe and sit around and find some place where you can be connected to nature or be connected to the microbes or the patients, whatever it is that you're interested in studying, and sit there with a journal or a pencil, something like that, and just observe everything around you. You know, we move really quickly. It's good to kind of stop and just look around for a while. Think about how maybe the flowers smell, or how hot it is out here, or whether it's rained or not rained in a while, whether you hear birds or don't hear birds, or whether there's shade. Think about all the kind of biology around you. And then choose a population that's around you and interests you, whether it's flowers or trees or insects or rocks. And think about something you might want to know about that population or group of populations. And write down what you want to study from that population, which will ultimately become your variable. It's oftentimes easier to choose the population first. That's why we kind of put this observation step first, and then the variables second. Doesn't have to be that way, though. What I recommend, though, is once you find a population you're interested in, think about something as far as your variables that will be simple to measure. Whether this is tree circumference or tree height, something like that. One easy measurement is counting things, since it doesn't even require any instrumentation. So now you'll want to take your population that you observed and were interested in, or groups of populations, and the variable that you think you might want to measure, and write that down into a testable question, something that is going to be simple and measurable, attainable, and specific. So limited to a particular time and place. You want to avoid why questions, not why are there bees here? That's not necessarily something that's testable. Instead, you could say, are the purple flowers outside of Sequoia attracting more bees than the white flowers, for example? So you've identified 2 populations, the purple flowers and the white flowers. and something you can measure, which is number of bees. You can sit there and count them. Finally, you're going to want to come up with a prediction. So I gave the purple flowers and white flowers and the attraction of bees as an example. Me knowing a little bit about pollination, I know that purple is a color that bees tend to key in on. So I might predict that the purple flowered species are going to attract more bees than the white flowered species. And so now I have pretty much a testable question that I can frame as a hypothesis. I hypothesize that the purple flowered species outside of sequoia in the month of October will attract more bees in the morning than the white flowered species. And so that's something that's simple, it's measurable, it's obtainable, and it's limited to a particular time and place, and it's not a why question. It's something I can go out there, test, and answer. So I kind of mentioned this a little bit. Counting is an easy one. Counting the bees is basically something you need to think about. What will your data collection look like to address your hypothesis? And mine would be sitting out there and counting bees and trying not to get distracted. In addition to that, you need to think about what statistical test you would use and whether that statistical test would allow you to address your hypothesis. Because if not, you need to rethink how you're collecting data and what your variables are. So it's always important to think about, okay, am I going to be able to use a t-test if I'm looking at count data and a categorical variable like flower color? Or would I need to do something more quantitative? And so it depends how you set it up. If I went out multiple days in October and every day I counted the bees that visited purple flowers and counted the bees that visited white flowers, I would have two nice columns of purple versus white from different observation days, and I could probably do a t-test on that. But if I did it all in one day, I would end up with just two pieces of count data, and maybe I'd have to do something like a chi-squared test that we haven't talked about in this class. So it's really important to kind of think about how you're going to answer your hypothesis before you go out and do it, because otherwise you might have to do it again. All right, so now it's going to be your turn. Your assignment before the next video is going to be to come up with a question that has clearly defined variables and can be framed as a testable hypothesis. So rather than rushing to the next part, what I want you to do is take 5 or 10 minutes and sit outside and think about your surroundings, or sit in the arc and watch all the other students. Again, students are a population, so that actually would work. Think about how things may or how they may not work. Kind of try and think like a scientist. And if there's something that sparks your curiosity, that might be where you want to go with it. It's really tough to do science on questions that don't interest you. So try and find something that, for whatever reason, is interesting to you. And once you have identified that thing, what I want you to do is come up with what your variables are that you might be interested in addressing that question with, and frame it as a testable question. And take that testable question and try to write down your prediction or hypothesis. We're not going to go any further than hypothesis at this point because we're going to talk more about experimental design in the coming videos. But for now, you're going to observe. You're going to come up with a question and variables that might let you answer that question, and frame it as something testable that can eventually, based on your predicted outcome, be placed into the context of a hypothesis.